Okay, so this is about stuff I'm really familiar with, and it's about quantifying uncertainties in big genome data. Yes, so first of all, this is about discovering genetic variants in the first place, and also about something, if I have time for this, but I doubt I will, about something else. But here is about data concerning structural variants, in particular deletions and insertions, where I have been specializing in the recent past. And I have all these tutorials given today, which perfectly introduced me anyway by Feridun and by Ben also. So I don't have to tell you like how to, in principle, discover structural variants, but to have everyone on the same page. Again, it's kind of I extract the donor genome DNA, I break it into fragments, I sequence the fragments, which gives me paired and reads here. I map the paired and reads against the reference genome, and what I get is, in reality, and not in terms of beautiful and easy schematics, this huge pile of paired and alignments. And this is data which is uncertain. It's not only big. I mean, you can see this immediately. You have about 1 billion of such paired and alignments. It's also uncertain, as you can see in here. As Feridun pointed out today, approximately 45% of the human genome consist of repeat elements. And this, for example, is a region which appears somewhere else in the genome as well. So this is the integrative genome viewer from the Broad Institute. Here is the reference genome, and this is a region which appears somewhere else also. And then all the reads that are broken out there also align here and the other way around. And you cannot trust any of those alignments anymore. If you would like to predict something, you have to. If you really would like to make predictions in these areas, you should handle the uncertainty coming with the data here. Okay, so what kind of uncertainties will I be dealing with in the following? It's first of all alignment uncertainty, and it's also typing uncertainty first. Alignment uncertainty. So reads, as seen in the slide before, reads can align to different regions, because these regions are repeat elements. So region 1 and region 3 are very similar, or even are near identical. And read 2 could stem from region 2 or region 4, because those two regions, again, are near identical. What you can do as of today is you can compute for each of the alignments a score. And then you can compute a Bayesian type probability for an alignment to be correct, and I call this P in the following. So this is just a Bayesian type computation here over the scores of the alignments for the read. There's also typing uncertainty. And typing uncertainty means that even if I can rely on an alignment to be correct, I still cannot really rely on the statements that it makes. So a little bit of a movie on how you can predict deletions. You have a region in the reference genome that does not exist in the sequenced genome. That is missing here. Is 12 nucleotides missing here. And then you break out the fragment. You sequence the two ends, gives you a paired and read. You align it back to a reference, which is a paired, gives you a paired and alignment. And then here, this is longer than the fragment here. The problem, however, is that I do know the length of the alignment. I just subtract the left coordinate from the right coordinate, the reference coordinate, but I do not know the length of the fragment because I missed to sequence the internal part, so I do not know how long it was. What I can do is I can study the fragment length distribution. I can come up with this at least by some, in the meantime, routine procedures. So I can now this is fairly normal. It's actually not. There's someone in the audience who can tell you all about this, Christopher Salin. But let's assume it's fairly normal. And then what you can do is you take the alignment length, and someone says that there is maybe a lesion of length 12 here, and then you figure out where on the x-axis your alignment length is, and then so assume it's maybe here, And the blue distribution is the one for the assumption that there is something. The red distribution is the one for the assumption this is just a regular region. There's no deletion of length 12. Then you put it here, and then you compute a probability, Bayesian type probability again, by comparing these two values. Like this is the value for the right distribution, and this is the normalization by summing up the two values for the two distributions. For example, if you put it here, then this value is this, and the red value, this one here, is this, and then just an ordinary Bayesian type computation. Yeah. Nothing really special. There's also split read evidence. This looks much easier at first glance. This is just the gaps in the alignment. You would like to make use of this information as well, but it's also typing uncertain about this type of information. Unfortunately, I mean, placing gaps in alignments is one of the oldest, is mostly maybe the most classic problem in bioinformatics. There's effects like gap wander and gap annihilation, where gaps merge or then disappear, or gaps wander, so misplaced by a couple of nucleotides. So you never can trust the gap in an alignment, and certainly the short read aligners haven't contributed to solving this eternal problem. Even for the more sophisticated alignment programs, this is still a considerable issue. So you never trust the gap. So there's also uncertainty about this kind of stuff. It works relatively fine for gaps of length at most 30. Still, there's uncertainty, but nevertheless, you can do it. But there's also this twilight zone of length, insertions and deletions of length between 30 and 150. I have to switch back to this slide before. Why is this actually? So 30 below 30, I can use the gaps, yeah, and beyond 150, I can make use of this picture here, because the blue curve is so far right out that I get a statistically strong statement, and then I can separate the alignments from the alignments indicating variance from the ones not indicating something. If I have to deal with a variant of length at least 150. Yeah? So in between, there is this twilight zone, statistical twilight zone. It's really hard to make predictions in there. You have to work a little harder to get something done there. So I did this. This is a long time ago. In the meantime, so we wrote this paper here, clever, click enumerator variant finder. It appeared in Bioinformatics 2012. It's actually the first paper. I also was told, like, now to talk about the applications of SnakeMake. My application of SnakeMake. So this is the first paper where I used it, and we were recognized by the faculty 1000 for that, and we are able to discover in fast time. Yeah, we're not the first ones to do this, but we are by far the fastest ones. And we are able to execute billions of statistical hypothesis tests in very short time, and then we could use this also in the frame of the Genome of the Netherlands project, which is a population scale project. So the fastness was the decisive factor here. Then we also combined this with a split read, never published, by the way, split read aligner, and we had another paper made clever, and this is what we used in the Genome of the Netherlands project. That's a long time ago. By the way, this is also in the classification scheme of Verdun. This is also able to handle multiple genomes, by the way. Yeah, so this is the Genome of the Netherlands. Okay, it's the German title. I patched together some slides here very quickly. Anyway, we had 769 individuals, and they came all in families. So 231 trios, 8 dizygotic twin quartets, and 11 monozygotic twin quartets. This yielded 769 individuals when we sequenced them all at fairly, not low coverage, yeah? Medium coverage, 12x. I think on average actually 15x in the end. fairly evenly distributed across the regions of the Netherlands, and then we sequence all the people, and this was the data we had to deal with. We applied SnakeMake, we mapped them, we discovered the variants in them using Clever or Made Clever or whatever, and then we did all the jobs we had to do. And this yielded this paper here, which is the main paper, appeared in Nature Genetics in 2014, and our contribution was this year. Here, this is the length range of the insertions or deletions. So minus is for deletions, and this is 30 to 150 here. And the blue plot is the 1,000 genomes plot, and this is the amount of variants discovered. And here in this region, nothing had been discovered by the 1,000 genomes project. And then in the frame of Genome of the Netherlands, we finally discovered lots. And this was our contribution to the project. So it's actually, well, someone said, well, it's not really a twilight zone anymore. We can make it visible. Yeah. So I may have to rethink the name in the meantime. Anyway, now the motivation is, Ben just gave a talk about like discovering somatic variants. It's a different business for certain reasons, for a variety of reasons. And now the idea is, if you go to a cancer variant database and you look for a twilight zone insertions or deletions, there's nothing. Nihil, nothing. Whatever. Yeah. So the idea was, okay, we know how to discover this. Why not fill up these databases too? However, there's a little more to this, as I said. So let's go back to this here. Let's go back to the problem. So this is somatic deletions and insertions. These are, you have a control genome from which the cancer genome evolved. And then you see variants in the cancer genome that had not been yet in the control genome. You still have the germline variants that already had been in the control genome in comparison to the reference, and the other way around for the insertions. You have new sequence only in the cancer genome. You would like to discover this. And then there's this problem of heterogeneity. You then want to determine, what you have to do is to determine the genotypes of the variants. If you would like to predict somatic variants, you have to do this. And the allele frequencies, the genotypes, the probability that the genome copy in the sample harbors the variant. And here in the healthy sample, we all have a mother and a father, so either both of them transmitted the variant, or only one of them transmitted the variant, or none of them transmitted the variant, gives you a probability of 1.5 or 0 for encountering a genome copy harboring the variant. In cancer, nothing is. It's totally unclear. Here, I presume to know this picture, but I do not know. So there are all these clones, and they come all with different sets of somatic variants. And if I then would like to know the probability that the genome copy in this sample here harbors a somatic variant, I can only assume nothing in some sense. I can assume, for the mathematicians here, I can assume it's a rational number because it's a finite number of cells, but that doesn't help much. So everything between 0 and 1 is conceivable. Yeah? And I would like to predict those allele frequencies both in the control sample and in the cancer sample. So, and what's the idea behind this for finally calling a variant as somatic? So I call like theta sub h is the allele frequency for the healthy sample, which is either 0, half, or 1. And theta sub c is the allele frequency for the cancer sample, which can be anything between 0 and 1. And this is data I get for the control genome here, H like healthy, and this is the paired data alignment, data which I get for the cancer genome. And then I would like to compute this likelihood function here. Given the data, what's the likelihood to encounter allele frequency theta sub h and theta c? Yeah? And in the end, if we have control of this function here, then we have to do this very fast, yeah? Because we would like to compute this integral over this function here, which is proportional to the probability that given all the data from the two samples, the variant in question is somatic. And we would like to do this for all the variants, for all the possible somatic variants I encounter in a step before. Okay, so now come the uncertainties, and this is the problem. First, assume we don't have any uncertainties. Still then, cancer is more difficult than healthy, because here, say, someone says here is maybe a somatic or whatever kind of variant, yeah? This is the reference genome here, and this is data from either the control genome or the cancer genome. So these are the pattern alignments spanning the INDEL, and these are the pattern alignments indicating gaps, which might indicate the INDEL, yeah? So assume we don't have any uncertainty, we know all these alignments are correct, so they indicate the correct placement of the reads. And we also know that those two here, the red ones actually, there's also no typing uncertainty. The red ones, they are associated with the variant allele, and the gray ones are not. I then can conclude that in control I had an allele frequency of 0.5, because the gray ones, they needed to come from the one ancestor and the other one from the other one. Yeah? So both of them, like they disagreed in transmitting the variant, obviously. However, in tumor, I can only say, well, this is maybe a maximum likelihood estimate for the allele frequency 4/7, because 4/7 of the alignments gave rise to the variant. Of course, it could be also different. In tumor, we only have a probability distribution over the different allele frequencies, which is proportional to the likelihood function. Okay, let's recall. Okay, so every alignment is subject. We assumed it was not like that. So let's go back to this. Every alignment is subject to alignment uncertainty. We have a probability, though, that for each of the alignments that the alignment was the correct one, and we have for each of the alignments we have a probability Q that it was associated with the variant if the alignment was correct. So and then this picture here gets a probability that this is the correct picture. Yeah. So the combination of alignments being correct and alignments being red, meaning being associated with the variant. So for each of the alignments 1 to 7, I have this probability. I can compute this from other considerations, and then I can compute the probability for this scenario. I call it phi 1. Yeah, this scenario here to be the correct one. Yeah, by making use of the pi and the qi and so on. Okay, could be also this picture, could be a correct one, and we also get a probability for this one. Yeah. So this is less likely, but it still could happen. Huh? And overall, now the big problem is I would like to compute this likelihood function really fast, but the big problem is I have to integrate over all the possible realizations phi. This is a big sum, and this is an exponential number of scenarios. Actually here, like it's 3 to the number of alignments, meaning an alignment can be either not correct, or it can be correct and not indicate the variant, or it could be correct and indicate the variant. So it's either not there, gray, or red, referring to the other picture. Huh? So it's and usually you have like about 30 to 40, 50 or so something, or even 60 or 80, 3 to the 60 or 80 many alignments, which is more than 10 to the 10, or I don't know what. Huh? It's a lot. And you have to do this for many loci in the genome, so you have to be quick. So definitely not do this here. We integrate in a different way. We make use of a latent vary model, which is the following. Someone also asked, I mean, this is the last talk of the day, but someone yesterday asked for more methods. Well, it comes a little late maybe, but yes. So here, this is the latent vary model. You have this is a model for either the control genome or for the healthy genome. So this is either the healthy allele frequency or the cancer allele frequency, and each of the columns represent an alignment. Yeah? This is the allele frequency. The circles denote latent variables, and the squared things, they denote things that you can observe here. Those are the paired and the spanning alignments. So this is the alignment length. Yeah? And this here are the split read alignments. So this is either 0 or 1, 0 for not having a split that indicates the variant, and 1 for having a split that indicates the variant. And say zoom in on say here, this here that well, omega is 0 or 1. It's 0 if the element is not correct. It's 1 if the element is correct. And this here it's 0 if the alignment is not associated with the variant, and 1 if it is associated with the variant. And for example, here you can infer that, given that the alignment is correct, omega I is 1, and given that it's not associated with a variant, then the alignment length is just distributed like the original fragment length distribution. Yeah? If the alignment was correct and the alignment was in fact associated with a variant, then the alignment length is distributed according to the fragment length distribution shifted to the right. I think there was a blue curve in the slide before. Yeah? This is the red curve and this is the blue curve. So things like that are in this model now systematically to be systematically evaluated. Yeah? Okay, so we wanted to compute this really fast. I keep repeating this. So we have the following lemma. We have the allele frequency, we have the variables for read pair information, alignment length, we have 0, 1 for binary variables for split read information, and we'd like to compute the likelihood that given all the split read data and the paired read pair data to see this theta. And we have the following lemma. Actually, this decomposes into just factors, which statistically speaking means that we observed a conditional independency of the data given the allele frequency, which is hidden. What we assume we assume we know, then we know the alignments are all independent. Yeah? So as a theorem, we end up with the following theorem. We can compute this in runtime linear in the number of alignments. So it's not 3 to the number of alignments. It's like just a constant factor times the number of alignments. And then also, it's easy to show that this has a global optimum on 0 and 1, which is a nice behavior. This is the maximum likelihood estimate. So it's a well-behaved curve, and you can evaluate the function very fast. So you can integrate. And then you compose two models. You have one for the healthy part. So Z is either 6X or Y. So these are all the alignments for the healthy genome. And these are all the alignments for the cancer genome. And by also observing dependencies between the healthy allele frequency and the types of the alignments in the cancer, this is for modeling impurity, yeah? Because we have a lot of healthy cells already also in the cancer genome. Yeah? This does a good job, by the way, if you do this. If you leave it away, we have worse results. Yeah? Not decisively worse, but if we do this and we have a good guess on impurity, results get better. So we can compute this finally fast. We can compute this. It's so fast that we can indeed here compute the integral over this function. And then we had the following. We had simulated data here, modeling some evolutionary kind of. We have a very good simulated data set with several clones having evolved and having emerged from a founder clone and so on and so forth. So we then have recall here, which is the number of somatic variants actually detected by us or by the other methods. And then precision is the fraction of predictions of ours or the other methods that are correct. And then if you vary the probability and filter out possible somatic variant calls by by having to satisfy that they have to have that the probability for them has to be at least a given threshold. By raising this, we certainly lower the recall because we filter out ever more variance. So the curve is going to the left. This is a natural. You cannot avoid this. However, I mean, also if the method is good, then the precision should go up. Huh? And this is what happens. And so the whole method works very well. And if you compare ourselves, in particular in the twilight zone here, between 30 and 100 we now have into here, we see there's no other method, by far no other method on market that can do a good enough job. And we now have a handle to do this. Yeah? This is certainly operable precision rates at still high enough recall. Yeah? You can also compare it with standard pipelines in action at the ICGC, for example. They have recall of at most 30% and precision. This is for the whole range of 10 to 100. They have recall of at most 30%. They don't have any twilight zone indels, yeah? But in the lower regions, 10 to 30, they have recall of at most 30% and precision of at most 50%. So somatic indel discovery is still in its infancy, I say. Huh? And with PROSIC, which is our method, we have recall of at least 60% and precision of at least 85%, which is this is an improvement. And the German Cancer Research Center has submitted a proposal to get us aboard here. Was just the wrong point in time to write this proposal because they're now actually writing up the papers and they don't want to get disturbed by us. So let's have a look. Maybe let us in at some point in time. Okay, motivation number 2, and this is I make use of the genotyping part of the model, so I only have to deal with healthy data, and I do this for the following purpose. You heard a lot of talks already, very good talks. We're dealing with genome-wide association studies. We have a cohort of people, and some are affected by disease, say, or by whatever kind of phenotype, and then you'd like to genotype all the people for lots of variants and then try to find variants that are associated with this phenotype. You don't want to sequence all these people. Possibly you would like to only genotype a restricted amount of SNPs by cheap array technology and then impute the genotypes of the other variants. Yeah? We learned in the very first talk of this meeting how to do this. And then we impute all the other variants, and then we have a lot more variants that you can possibly associate with the disease, which is very helpful. Okay, so this is just a picture. You measured. Here is your cohort, and this is the reference data. You have a reference panel in the background for which you know all the genotypes of all the SNPs. Yeah? And then you measure SNP 1, 3, and 5, say, by cheap array technology for the cohort. This is the lower part. And then you try to predict the other ones, which is called imputation. And these are then imputed by making use of statistical models that allow you to infer those by making use of the genotypes down here in combination in relation to the genotype combinations up here in the reference data, in the reference panel. And the novelty here is now that we got the structural variants integrated into the reference panel, and we do the same thing then, we impute them. So we start out with only, we now have this reference panel for the genome of the Netherlands. Yeah? When we have all the structural variants we discovered, we have them integrated in here. And then we, given just the SNP status for the people of a cohort, cheaply evaluated by array technology, we can predict those missing structural variants. You really don't have to sequence any of them. And this works fairly well. If we then, well, this is just some pictures. This is a redundant slide, I think. It looks very similar to SNP imputation plots. Let me go there. This is actually conditionally accepted at Nature Communications now. This year. And if you compare also the 1000 Genomes project have issued a reference panel as of lately, but well, I can claim it's worse. So this is the percentage of wrongly imputed discordance is the percentage of wrongly imputed. How can I explain this? So we had a ground truth, and we could like benchmark against the ground truth, which we inferred from real sequence data, and we did as if we didn't know, and then we imputed, and we compared the imputed with the sequence data. And then the discordance is the fraction of genotypes that were wrong in comparison with the ground truth. Huh? And then, for example, in particular, this part here, where we only consider the alleles that have, well, using this which have low allele count in the reference panel, yeah, above a below average allele count in the reference panel, then we see that this is on. Sorry, what is this? Actually, this is for deletions, yeah, and then we predict the deletions. We impute the deletions at a rate of 95.5% accuracy. And we miss out only on 28.5% of them. So we discard 28.5% of the deletions. We say we cannot make predictions with them because the data here is too uncertain. But for those, we predict them fairly well. And this is the scores for 1000 genomes. They miss out on 41.3% and predict them at below 90% accuracy. So for mobile element insertions, the scores are even better for us. In comparison to 1000 genomes, they have to discard nearly 2/3 of them before they say, well, we would like to do something, and it's still like 30% of them are wrong. On top of that, we have all the twilight zone indels in our reference panel. 1000 genomes doesn't. We also have complex indels, yeah, and we have duplications and inversions, and all of that is not necessarily in the 1000 genomes panel. Yeah. We also have like, well, this is not so interesting maybe here, but yeah, we have 150 deletions. We discovered them to be in materially and statistically significant in LD with disease SNPs discovered before. And well, 2 of them had already been published in Nature papers or something. So 130 more to explore. Maybe they're the cause of variance, I don't know. I'm not an expert in this. And we also control the false discovery rate in the procedure we applied. Okay, so I think I have an early end today. I would have another third part. Well, let's summarize a little, at least. Let's summarize a little at this point. Like in current generation bulk sequencing, you have alignment uncertainty. This was what I was talking about today, so far. You have read placement uncertain. You have the read placement uncertain, and you have the association with the variance uncertain. In third generation reads, many people think, okay, fine, we get rid of a lot of problems with this. Yes, alignment uncertainty will decrease. However, typing uncertainty will possibly even increase because of the higher error rate. So not yet, these uncertainty considerations are not yet to be put away. Yeah? So there's still something to be done. And then also single cell sequencing, which is kind of a big topic. I'm now have become recently really interested in doing this because you have a lot of more uncertainties coming in, kicking in, like a lead dropout, or you always implicitly make the assumption of coverage uniformity here, which is kind of given a number of genome copies, then with equal probability I draw each of the genome copies. However, in single cell sequencing, this assumption is no longer justified. You also have the amplification errors, which can disturb the whole procedures quite a lot. Actually, still, the discovery of somatic SNPs in single cell sequencing is still really poor, to my understanding. Yeah? So even for SNPs, things do not really work out because of all these layers of uncertainty. yeah, that's about it. I skipped this part. I would have had another interesting story about testing 2 times 2 association, where, I mean, many people take the data there for granted, and they fill the 2 times 2 tables and take each data point and put a 1 here, put a 1 in one of the 4 possible cells. But if there is uncertainty about this data, then you have to deal with a probability distribution for each of the data points over the different cells. Yeah? And then we ran some simulation studies, really preliminary work here, and if we introduce a little bit of noise and then try to predict stuff, we found a lot of associations, disturbingly many, but I'm not sure whether I still have time to tell this. I'm also not, this is not really a positive story. Maybe I don't tell it now. Still to be explored. Maybe it's not so harmful, but I think in some cases it should make sense to try to deal with these uncertainties as well. Okay, that's it. Thank you very much.